Buenas tardes y bienvenidos un día más al Seminario Junior. Como sabéis, tenemos café, infusiones y bollitos que podéis coger ahora o bien después de la presentación de SERGEI. Están patrocinados por el Instituto Gregorio Millán Barbani y bueno, podemos aprovechar luego también para hablar sobre el seminario. También tenemos redes sociales, os instamos a que nos sigáis en ellas. En ellas vamos anunciando también los seminarios que vamos a dar y vamos poniendo fotos de los speakers. Con nosotros tenemos hoy a SERGEI, que es también un organizador del seminario, compañero, y para el que me gustaría pedir un aplauso porque acaba de terminar su doctorado hace menos de un mes.
Gracias.
Nos va a hablar de biopelículas, nos va a hablar de bacterias, así que, bueno, sin más dilación os dejo con él.
Vale. Muchas gracias. Yo voy a hablar en inglés. This talk is called Volume II because a few years ago, in the beginning of Seminario Junior, there was a first talk about biofilms, which I gave. But I suppose that almost nobody remembers about the talk. But don't worry, because it will be mostly a prequel than a sequel of the talk. So today I will tell you about what are biofilms and where we can find the structures. And next we will consider a different approach to simulation of biofilms. We start from the most simplest model which simulate how biofilm grows and how thickness of the structure changes. Next we will consider how we can simulate mechanical properties of biofilms. And finally we will talk about morphogenesis and formation of wrinkles in biofilms. It is exactly what my PhD thesis was related to. So, when we talk about bacteria, usually we can imagine this genetic tree that of all species in Earth. And bacteria are prokaryotic species. It means that it's cells without core, without kernel, in difference with eukaryotic species, which are plants, animals, and us. And usually, so me, for example, at the beginning of my work, I imagine that there are more variety of complex and multicellular organisms than simple unicellular organisms as bacteria. But in reality, it's not true. And real genetic tree looks like this. Here, red zone are eukaryotic cells, which include not only all multicellular life, as trees, animals, and fungi, but also unicellular organisms. And all blue part are different bacteria. So it means that more than 3/4 of all species on Earth are actually bacteria. And genetic difference between different bacterial species can be larger than between human or mushroom, for example. Besides, most of bacteria do not live as solitary cells, but organized colonies known as biofilms. This structure can be found everywhere on Earth, from volcanic geysers to subglacial lakes. They can be very large, as these biofilms which cover the bottom of a lake or stream in Yellowstone National Park. They also can be organized quite small structures, as this mushroom-like structure, which also grows in some stream. And we can also find biofilms in experiments, as these two colonies of Pseudomonas aeruginosa and Bacillus subtilis, which grows in Petri dish and is famous for ability to form complex wrinkling patterns, which also used by colony as a network of channels to transport resources. Despite this variety, usually formation of biofilm follows the general scheme. It starts when a few planktonic cells can be from different species attached to the surface. Next, bacteria change their phenotype, lose motility, and start to consume nutrients from substratum below and substrates in bulk fluid to produce extracellular matrix. And extracellular matrix is a mixture of proteins, extrapolysaccharides, extracellular DNA, and water, and it's the main feature which differs biofilms from isolated cells. The matrix can hold cells together and prevents washing them out from the flow, so it provides stability to bacterial colony. Besides, it prevents penetration of antibiotics inside the colony, so it protects biofilm from aggressive environment. And finally, it facilitates cell-to-cell communication and horizontal gene transfers, so that biofilm behaves exactly indeed as community of cells where they can separate their functions and when biofilm is large enough, then spore cells are released to colonize more surfaces. Biofilms has a very huge impact to all human activity. In many applications, they are undesirable because they can cause infections, colonize processes and invalid devices, and also cause biocorrosion. An important example is cystic fibrosis. It's a genetic disorder which makes lungs vulnerable to colonization by Pseudomonas aeruginosa biofilm. In this photo, you can see lungs of a patient with cystic fibrosis, and you can see that the right lung is almost completely colonized with bacteria, and so this person probably have problems with breath. And generally, for this disease, bacterial infection is responsible for 85% of lethal ends. Another example is biocorrosion, which can destroy construction and value in water, and or also leads to energy losses for different industrial applications. However, not all activity of biofilm is harmful. They can be apart from spreading disease. They can also protect us from different infections and facilitate the digestion process. They can be used for wastewater treatment in this, as for example, in this trickling filter, where we have usually in this filter we have a mixture of rocks and sand and water flows through this mixture, and there are a lot of bacterial colonies on sides of rocks which can also use it for remove waste from water. And biofilms also can be used for fermentation processes in food production, for example, in bioreactors, as you can see in this picture. In order to prevent formation of biofilms where they are undesirable in these first two applications, and on the contrary, increase their growth when they are beneficial, we use methods of mathematical simulation. And the first and probably the oldest approach will be considered now. It's simulation of biomass growth. So the first model was proposed in the 80s. It was 1D. And here we consider biofilm which contains n different species of biofilm, of bacteria. Fi is volume fraction of each species, so sum of these volume fractions is obviously 1. And this biofilm grows in a mixture of m different substrates from which bacteria can take nutrients to survive and produce extracellular matrix. In this 1D model proposed by Warner and Gudger, we have 4 main components. First one is production of extracellular matrix due to utilization of substrates. Second part is expansion of the boundary of biofilm. Third part is diffusion of substrates, well, in bulk fluid and also inside the colony, inside the biofilm. And the last part is the possibility to attachment and detachment of bacterial cells due to external flow. Well, this model includes few main equations, which are actually quite simple. First one is transport of biomass. We have that volume fraction, production of volume fraction of fraction of bacteria is proportional to the present amount of these species. And also we have additional terms which represent emergence of bacteria due to internal flow in the biofilm. The second part is diffusion of substrates. So we have usual diffusion equation for substrates, but we have additional term which represent utilization of the substrate by bacteria. And the last part is equations for the boundary of biofilm. We have that the thickness of biofilm, or the upper boundary, grows proportional to velocity of production of biomass, and also we have attachment and detachment term. So in velocity is of course a derivative of velocity respect to vertical coordinate equal to average growth of biomass. So it means that the higher is growing rate of bacteria, the faster the boundary will spread. This biofilm, even though this model is very simple, it can provide some useful results for bioreactors or filters. An important case is well, the test case of unrestricted growth of biofilm. So in this case, we have enough nutrients for infinitely thick biofilm, and we just try to analyze how it will change with time. So we consider two different species of bacteria, one heterotrophic, which use organic products and oxygen to grow, and another autotrophic, which needs only oxygen to produce biomass. And also we have inert biomass, which represent products of cellular activity and dead cells. So we start from very thin biofilm, and firstly we can note that we have an interval of exponential growth, which corresponds to the case when there are enough of nutrients and resources for production of biomass. So in this case, we have homogeneous distribution of all species along the thickness. So in the interface, we have the same volume fractions as we have at the bottom of biofilm. So here it's drawn like we have some distribution along this direction, but actually all species are spread homogeneously along this line. Well, this fraction proportional to the thickness of these rectangles. Next, there is not enough nutrients and enough oxygen, and the growth of biofilm is slowed down, and we have only linear growth of biomass. Until the moment, so in this moment, there starts a competition between heterotrophic and autotrophic species for the oxygen. And well, since we have mostly we have heterotrophic species from the beginning, then at some moment all autotrophic species disappear from the interface of biomass. So it means that the higher layers of biofilm, where we have more nutrients, more oxygen, and more resources from bulk fluid, is completely occupied only by heterotrophic bacteria, whereas inside the biofilm we can find autotrophic one, which requires only oxygen to survive. And next we just have continuous of this. So this behavior can be. We have similar behavior for the later growth of biomass, and finally we have that on the outer layer we have only heterotrophic bacteria, and inside we have different species of bacteria. This phenomena is well, is quite important because it means that we have a distribution of different species of different way of life for bacteria on the outer rim of biofilm and inside the colony. And if we, for example, want to treat with structure with antibiotics, we have to use different antibiotics for the bacteria outside and inside the colony. And of course, all cells inside has a higher tolerance because antibiotics should have to diffuse inside the colony, which also is slowed down by the biomass. So it means that almost in any case when we try to remove biofilm with antibiotics, we will always have a few cells which lives inside the colony, which cannot be removed, and in few days the biofilm will be growing to the same size again. So it means that if we want to remove it from, for example, processes, the patient has all the time use antibiotics to prevent emergence of biofilms on the processes. But biofilm is not always one-dimensional and homogeneous. Sometimes we have a very complex boundary, and it means that this model have to be generalized to 2D and 3D cases. A possible generalization was proposed by Docker and Clapper, and it has a little bit different model. For production of acetyl matrix, we use Darcy's law, which is true for viscous and compressible fluid. So we consider biomass as viscous and compressible fluid. For So inside the biofilms, the velocity is given by gradient of pressure. It means that the production of extracellular matrix pushes cells and provides movement of the boundary. Outside the biofilm, we always have zero pressure, and we have two reaction diffusion equations for the substratum. So outside we have one diffusion coefficient, and we don't have any utilization of substrates. And inside the colony we have utilization of substrates and another diffusion coefficient d2. Finally, the velocity of biofilm can be given similarly as we did for 1D case. We say that divergence, so in the 1D case we had just derivative, here we have divergence, and divergence of velocity is given by production of growth or production of extracellular matrix, which in this case we set equal to utilization rate of substrates. It means that all substrates used by bacteria is used to produce extracellular matrix. The last question is how to simulate movement of boundary, because in this case we have free boundary problem. We had to set boundary conditions on the interface between fluid and biofilm, but we actually don't know where this interface is, because boundary is also part of the solution. One possible method is the Levelset method. Means that we define a Levelset function phi, which is positive inside the colony, in negative outside the biofilm, and the boundary is given by zeros of this function. Advantage of this method is that we can use the same, for example, if we use finite element method to resolve this problem, we can use the same mesh to all the time and just combine all our properties as concentration of substrates and biofilm with this function. The disadvantage is that this method is implicit and we have to solve additional partial differential equation to find function phi. And this equation is just a transport equation where we have derivative of this function respect to time plus transport of this of gradient of this function with velocity of production of biomass equals 0. And another condition is that gradient length of gradient of this function is always 1. It is necessary to make function phi Levelset function equal to signed distance to the boundary, because during solution of this equation, the change of the levels can be different, and we want to always to maintain it in more or less the similar rate. It means that, for example, after we make a few iterations, it can be that this function is 0 on the boundary, it grows very fast outside or very slow. And we want that this function always grows to more or less the same rate to apply our method. Well, using this method, Clapper and Dockery resolve the equation from the previous slide for 3D case and obtain more or less similar behavior to that 1D case. We have here only bacteria represented by green color and inert biomass, or dead cells represented by blue color. And we can see that inside the colony where there is a lack of oxygen and nutrients, we have mostly inert biomass, and all bacteria are concentrated on the outer rim where we have enough of resources. And besides, we can see formation of finger-like or mushroom-like shapes, because it allows bacteria to increase surface on the top of the colony and therefore absorb more oxygen or more nutrients. Another possible way to two-dimensional, multi-dimensional simulation is to use discrete model. One is cellular automaton proposed by P.C. Renau. And in this case, we divide all our domain to a square cells. And in each cell, we resolve diffusion equation for substratum and equation for the concentration of biomass. And also we set an occupation state small c, which is 1 for zone occupied by bacteria and 0 for zones outside the biofilm. So inside each cell we solve system of two quite simple differential equations, And the last step is how to update this occupation state C. And here we use cellular automata, and cellular automata usually follow few simple rules. The first one is if amount of bacteria, of biomass, in cell achieves some critical value C max, we have to remove half of all biomass to a neighboring cell. If there is at least one free cell, then everything is quite simple. We just remove half to biomass to one of those free cells. However, if there is no free neighbors, we have to choose randomly one of neighbor and move half of biomass there and repeat algorithm for that cell. So it allows us to simulate spreading of the boundary of biomass of biofilm without introducing some additional functions as Levelset function, and basically follow few quite simple rules. The disadvantage of this method is, well, as usual for discrete method on cellular automatons, that the solution is stochastic, and usually we have to calibrate our model, for example, parameters as C max and size of cells, using experiments. In all previous models, we consider biomass only from the biofilm only from the point of view of concentrations of bacteria and substrates, and did not take in account that biofilm can be considered as a thin film as has some mechanical properties. And in this section, we will try to consider which mechanical property can have biomass and can have biofilm, and how we can simulate them. Firstly, we can note that biofilm are very complex. And measurement of stiffness and Young modulus of biofilms is quite challenging. Firstly, it happens because biofilm is quite soft. It's softer than mostly material we use in industry. Secondly, it has heterogeneous structure, so it can be seen photo from atomic microscope for biofilm. So we can have biofilm usually have some porous structure, and is actually a combination of bacterial cells embedded into a self-produced matrix. And finally, that biofilm alter in time, so it means that if we measure properties for young biofilm, it can be different properties than for mature biofilm or for large biofilm. So this is why we want to have some model how these properties can be measured and how we can see the evolution in time and due to bacterial growth. Well, this photo and the idea of how the structure, of biofilm is, can be inspired for this immersed boundary method proposed by Hammond and Stotsky. The idea is to simulate biofilm as a framework of rigid bacterial cells connected by elastic spring and embedded into viscous fluid. So viscous fluid is, of course, bulk fluid on substrates outside the colony, but also extracellular matrix, which we have around each bacterial cells. So the behavior of the fluid and the system is given by motion equations for each cell and Navier-Stokes equation for viscous and compressible fluid, where viscosity of fluid mu depends on the position. So inside the biofilm we have low viscosity coefficient because it's just water or some surrounding fluid, and inside these green zones we have higher viscosity because it's already biomass and because it's extracellular matrix. And another thing that is external force F, which comes from each bacterial cells. So we have to update it depending on movement of these cells. And also, of course, movement of bacteria depends on the flow in the bulk fluid and the biofilm. And using this method, Hammond and Stotsky can estimate, could estimate mechanical response for biofilm in experiment for when we have biofilm growing in between two plates, and we share upper plate respect to the bottom one. And so, of course, biofilm has some mechanical response to this deformation. and using this method, we can calculate this response. And in these papers, the response coincides with experimental data. So it means that this approach can be used to simulate mechanical properties of biofilms and to predict how they can be evolved when the colony grows. And the last part of this talk is how to simulate morphogenesis of biofilm. Because mechanical behavior, mechanical response of biofilm can also be responsible to formation of bacterial shape. It can be seen, for example, if we have a usual example in industry like buckling, when we have a thin film and we apply forces on the both sides, then finally this film buckle into some non-flat shape. And the similar happens with biofilm, and it explains formation of wrinkles and folds, which can be seen in experiments for Bacillus subtilis, Metamonos aeruginosa, for example. Usually, so in this part, we will consider a little bit different part of type of biofilms, which grows on interface between agar substratum and air, and all nutrients will be absorbed from the substratum below, not from bulk fluid outside the biofilm. For this type of biofilm, was demonstrated that ECM extracellular matrix can be simulated as elastic media, and also there was a hybrid metal proposed by Espesso. They use cellular automaton behavior to explain how bacterial cells behaves. Next, they used usual reaction diffusion equation for distribution of nutrients and oxygen inside the biofilm. And finally, they couple both these methods with thin film approximation to simulate biofilm itself and its wrinkles. In this case, source of wrinkles was given by internal growth, and it was quite complex function, so it quite obvious that it can provide some complex wrinkling patterns, because we set very complex heterogeneous force, and of course we have heterogeneous deformation. However, in this paper, was demonstrated that wrinkling pattern can be obtained even for quite simple growth. For example, for homogeneous radial growth or azimuthal growth. And well, in this picture you can see how Pseudomonas aeruginosa colony grows and how wrinkles form on this colony. So we can change that we can see that their frequency changes, and that mostly this formation, this shape, is a result of mechanical instability of the biofilm. Why wrinkles in biofilms are important? Well, firstly, as I already told, that they can be used for biofilm use it as a network of channels. And in this picture, you can see that if we set some colored liquid in the agar substratum, then this liquid will be distributed along the colony using this network of wrinkles. And it is quite beneficial for biofilm, because usual transport using diffusion is very small. And if biofilm used only diffusion to transport of nutrients, it would not be very large, because it always would be around the initial configuration of biofilm. However, channels allows to transport nutrients for a very large distance, so it means that biofilms which can form wrinkles usually have a larger colony than bacteria which lacks these properties. And another importance of wrinkles is that they are related with defensive mechanism of bacteria. For example, Asali demonstrated that patterns of dead cells coincide with regions where we have wrinkles. And usually, for example, for Bacillus subtilis, patterns of dead cells are related with bacterial cannibalism phenomena, which means that if bacteria is treated by antibiotics or face some hazardous environment, some cells change their phenotype and start to produce toxins which kills another cells and use their material to enhance production of ECM. So bacteria tries to increase production of extracellular matrix to cover cells from antibiotics. So it means that if we have the formation of wrinkles can contain information about development of history of the colony. And also it contains information about mechanical properties of the colony. So wrinkling is not typical only for biofilms. It can be seen in many different applications. For example, the most usual one is crumpling of paper. There we can see some chaotic wrinkles and also organize it as an origami, for example. Wrinkles can be used in microelectronic devices where we use also bilayer structures. For example, some soft layer with thin metal sheet attached to it, and when and changing of usually thermic enlargement of the metal provides formation of wrinkles. And also wrinkles can be very large. For example, wrinkles in Earth's crust, like mountains, and change of thickness, is also can be described by similar model, because Earth's crust is much thinner than radius of Earth, so it means that we have a very thin film comparing to its size in other directions. And of course, wrinkles are very common in biology. It can be seen in animals, in vegetables and plants, and finally in biofilms, which we will consider now. Formation of wrinkles is also interesting because it's related to geometry. It's a mixture of mechanics and geometry. Basically, it can be described by Gaussian theorem egregium, which says that Gaussian curvature of surface is invariant of under local isometry, or in other words, curvature of surface is invariant under local isometry. Curvature is a multiplication of two principal curvatures, which are inverse to radius. And for flat film, for a sheet of paper, for example, it means that if we have initially flat surface and we transform it without stretching or compressing, so under local isometry, its curvature will be 0 everywhere. And it can be seen, for example, in origami, that if we fold a sheet of paper, then of course it's not flat, but in each face it's still flat. And the curvature is different from 0 only on very small regions of bending, of folds. And well, this theorem is true for infinitely thin films, but of course all real film has finite thickness, and it means that near these edges we will have very large stresses, and which can even cause collapse of material. For example, if we fold a sheet of paper many times by the same line, finally we can break it easily by this line. It happens because large stresses provokes collapsing of the material. In our case, Gaussian curvature is not 0, but it is provided by growth. And so now we will consider how growth can provide non-zero Gaussian curvature and therefore bending of thin film. Well, in elasticity and deformation, we usually have reference configuration, initial configuration of the biofilm, and it transforms to the current one. If we have deformation is given by deformation tensor f, which is derivative of new coordinate respect to old one. And also it can be given in terms of displacement field u. It means that we have some displacement for each point. We assign a displacement, and adding it, we obtain the final configuration. If we have growth, we have additional configuration, which is called virtual, because this configuration may not coincide to any displacement. For example, in this case, we have azimuthal shrinkage. We have that this circle shrinks azimutally, and therefore a sector disappears. And there is no displacement field which corresponds to this configuration, because, for example, if we consider this line, which transforms to this gap, it's not clear to which point should go points of this line, to the upper side of the gap or to the bottom. It means that it's not a function in any sense, because this point has two images. And here appears emergence a deformation, elastic deformation tensor, which purpose is to close this gap. And if we glue sides of this gap, it is easy to see that flat circle transforms into a cone, which is current configuration final solution. So it means that if we change distance inside the reference configuration according to some growth tensor, we finally can obtain non-flat configuration. And it's important to distinguish two types of growth. One is compatible when virtual configuration is possible and then it coincides with current one. For example, homogeneous isotropic growth is compatible because it means that each points just move further from the origin, and in this case we don't have any wrinkles. And well, in case of a compatible growth, we almost in any cases have wrinkles, except the case when we have very large frequency and the thickness of biofilm does not permit this type of wrinkles. Well, now we know how to obtain a cone, but how this cone can be transformed to a complex wrinkling pattern. The key is in interaction with the agar substratum below the biofilm. The shape produced by cone, produced by geometrical transformation, is not unique because you can see that cone looking up and cone looking down are the same configuration. If we glue sides of that gap, it can be cone which tip on the bottom and tip on the top. And we have also more solutions because, for example, we can consider a combination of these two cones. So outside we have cone which looks up, and in the center we have a cone which looks down. So we have infinitely many solutions because we can break this cone at any point. We have to choose only one solution from all this set. And we can do it because we have additional terms. Firstly, we can remember that biofilm has finite thickness, and it means that very sharp bends are not permitted. So bending energy of biofilm, of the material, try to make it smoother and avoid any folding and wrinkles. On the other hand, we have agar substratum connected to biofilm. And of course, large deformation, large displacement, as in these two cases, leads to large deformation of the substratum. So substratum try to avoid large amplitude and move everything to high frequency solution, but with very small amplitude. That means that final solution, the general shape is given by growth or by geometry, but it should be smoothed using bending term. So for thick biofilm we have only very smooth solution, and for thin one we can have a lot of wrinkles. And on the other hand, it have to the amplitude also have to be reduced due to interaction with the substratum. And the final solution is a balancing between these two different forces. So we try to simulate the biofilm using considering as a thin growing film. We can so if we don't have any substrate and we have radial growth, so it's the same as azimuthal shrinkage, we obtain the cone solution as I demonstrated before, and we can also see that the tip of the cone is smoothed because bending energy there goes to infinity. For azimuthal growth, we have a saddle. It's also possible to imagine that azimuthal growth, we have to add a sector to a circle, and if we try to remove it, to get rid of overlapping, we'll obtain a saddle. And if we add interaction with the agar, then we can see that amplitude of deflection decrease and frequency of wrinkles increase, because here we have already combination of two cones, and here we have combination of many saddles. So these examples were quite simple for quite simple growth, but if we set a little bit more complex growth, we can obtain patterns quite similar to one which we can see in an experiment. First one is heterogeneous growth when we have conical solution center and that saddle solutions on the boundary. So we have concentric circles, concentric wrinkles in center, and this rosette-like shape on the boundary. Here we have large growth on the outer rim and small growth in center, and we have mixture of these different wrinkles. We have more channels outside than inside. Here we have homogeneous and isotropic growth, but interaction of the substrate allows obtain wrinkles even in this case. And we can see that we have a lot of wrinkles in center, near origin, and they decrease going to the boundary. And the final example is homogeneous growth, but heterogeneous biofilm. The center of biofilm is thicker than the outer rim, which is quite logical because biofilm starts growth from this region, and so here we have more biomass than outside. And we can see this circle, which can also be seen in many experiments for Bacillus subtilis biofilm, for example. So, to conclude, so I would like to say that biofilms are the most widely spread life form on Earth, and they have very huge impact on our activity. And this why simulation of biofilms from mathematical point of view is quite important. And besides, simulation of biofilms is quite challenging because we have a lot of different processes. In this case, I consider only as from the point of view of production of extracellular matrix and mechanical properties, but also there can be simulation of cell-to-cell communication and quorum sensing, which is the interaction between different cells of bacteria. And so this field is quite interesting from the point of view of mathematical simulation. And also that we have to combine many different approaches to obtain a correct model, a good model, which includes a lot of different features which we can see in bacteria. And thank you for that.
Pregunta.
Okay, Sergei. So in the slide 27, yeah, I see like two patterns. First, you can choose in the center of your biofilm to be radial or outwards or something like that, and then to be bigger on the inside or the outside. Is that controlled by two parameters or something like that? How can you control that?
Well, here the growth is heterogeneous.
Okay.
It means that the center grows differently than outer rim.
Okay. So what controls
in first two examples. And well, here center grows radially, and outer rim grows azimutally. So we have different patterns. In this case, center grows just slowly than outer rim. They both grows isotropically and homogeneous. Well, yes, grows isotropically, and inside each region is homogeneous growth. But outside is 2 times faster than inside. And it gives us these wrinkling patterns. In this example, growth is homogeneous. But the difference is that here we have interaction with the substratum. So even if we have homogeneous growth and it tries to expand, it leads to large deformation of the substratum because it also forces points of the substratum to expand. So in this case, it prefers to wrinkle and maintain the same area. And in this case, we have different thickness. So center is thicker, and it prevents formation of wrinkles in center. And all wrinkles goes to outside, and here we have this corona-like shape.
Okay, and are these all the patterns you can see?
No.
Okay.
Well, you can see you can set different types of growth. Idea is that here we actually set quite simple types of growth because, well, it's combination of 2 quite simple types of growth. Or in this case, it's even homogeneous, and we already can see complex patterns. So it means that probably that well, it's heterogeneity comes not from the high heterogeneity of growth, but from the geometry and mechanics of this process. So it's sort of that not each wrinkle is developed by only that region. Like not for each wrinkle we have to set some specific behavior of bacteria around this zone, but we can set general behavior for all the domain, and it will wrinkles, and it wrinkles in quite complex structures.
Okay, thank you.
¿Hay alguna otra pregunta? ¿No? Pues antes de despedir a Sergei, Quería aprovechar para recordaros que el próximo viernes va a haber un seminario. Debido a que tocaría el 8 de marzo y hay una huelga convocada, se ha movido ese seminario para el 1 de marzo. Sigue siendo en este aula, sigue siendo en la misma hora, pero hemos movido la fecha, ¿vale? Así que, si no hay más preguntas, agradecemos de nuevo a SERGEI su charla.